So as previously stated, the central nervous system is the integration and command center of the nervous system. It consists of the spinal cord and brain. It is estimated that the human brain alone contains over 100 billion neurons and significantly more neuroglial cells. The adult brain can be subdivided into four basic regions: the cerebral hemispheres, diencephalon, brainstem, which includes the midbrain, pons, and medulla oblongata, and the cerebellum. Each of these structures have specific and overlapping functions critical to overall organismal function. For instance, the structures of the diencephalon include the thalamus and hypothalamus. These structures act as relay stations that organize incoming neuronal signals by category, regulate autonomic nervous system and emotional responses, as well as regulates a number of homeostatic reactions. The medulla oblongata contains a group of nuclei. If you remember, nuclei are collections of cell bodies in the central nervous system. The nuclei in the medulla oblongata include controlling neurons of the cardiovascular, respiratory, digestive, and other critical systems. From an evolutionary point of view, the cerebral cortex, also known as the neocortex, is the newest portion of the human brain. It is where the conscious mind resides and is responsible for higher thought, memory, and the initiation of voluntary movement. It accounts for 40% of the brain's mass. Its convoluted appearance nearly triples its surface area. The elevated ridges of the cerebral cortex tissue are called gyri. The gyri are separated by shallow grooves called sulci. Sulci separate the brain into distinct functional regions known as lobes. They are the frontal, parietal, temporal, and occipital lobes. There are two cerebral hemispheres. A deep sulcus known as the longitudinal fissure separates the right and left hemispheres. As previously stated, neuronal activity is a high metabolic activity. It therefore requires a nearly continuous delivery of nutrients via the cardiovascular system. This picture is a medical image of the large and extensive network of blood vessels responsible for delivering blood to the brain. The spinal cord is responsible for the transporting of neuronal information from the brain to the periphery and from the periphery to the brain, as well as the integration of neuronal signals. We have already reviewed the anatomy of the spinal cord and how it mediates reflexes and sends signals to the brain and periphery. You should now be familiar with the basic functions and locations of the labeled regions of the spinal cord. Given its importance, it is clear that the central nervous system must be well protected. Most superficially, the entire central nervous system is encased in bone. This is accomplished by the vertebrae and the flat bones of the skull. Just deep to the bony protection are the meninges, 3 layers of connective tissue. The most superficial layer is called the dura mater. It is a fibrous collagenous tissue. Just deep to the dura mater is the arachnoid mater. The arachnoid mater is a loose covering separated from the dura by a narrow serous cavity called the subdural space. The deepest tissue is called the pia mater. It is a delicate connective tissue heavily invested with small blood vessels. The space between the arachnoid mater and the pia mater is referred to as the subarachnoid space. This space is filled with circulating cerebral spinal fluid. Cerebral spinal fluid forms a liquid cushion that provides buoyancy to the central nervous system structures. The brain, for instance, is suspended in cerebral spinal fluid. The buoyancy effectively reduces brain weight by 97%. Think about how you feel lighter when you are immersed in water. Cerebral spinal fluid also helps nourish the brain. Finally, the brain is protected by the blood-brain barrier. Though levels of ions, hormones, and amino acids may vary within limits throughout the body, a much more constant environment is required for brain function. The blood-brain barrier provides extra protection, acting as a barrier to diffusion and substances entering the central nervous system. The blood-brain barrier consists of the endothelium of the capillary wall, a thick basal lamina surrounding each capillary, and the pseudopods of the astrocytes. These three structures limit the types of substances that may enter the central nervous system.